NGS data, like any other science project, also has issues of data quality. So let's see what issues are there. The issues can be, there can be vectors, adapters, we can have low quality regions, low complexity regions, contaminants, as well as duplicates. Should we keep them? Depends on your question and depends upon how much it affects your downstream analysis. So most of the cases, it's not the case that you should be okay with that. There are two kinds of softwares that deal with these quality issues. One of them is filters and the other ones are classified as trimmers. Filters, they throw away the whole reads as long as if they are not satisfying the criteria, whereas trimming softwares, they clip some specific regions of those reads. So in this way you can have some partial reads still available for you. Vectors may not be common in NGS data analysis, though they were there with the Sanger sequencing reasons, because in the NGS machines there is no cloning, so cloning step is skipped. But still we have two approaches to handle those issues. If you are doing Sanger sequencing, you can deal them by LUCI or UNIVAC. LUCI is good for already known vectors, so what it does is it looks for the cloning sites for the given vectors and recommends how to trim the sequences. So it's kind of doing pattern finding jobs, and then gives you an idea where we can have those regions coming from those cloning vectors. UNIVAC is good for unknown vectors, so it performs BLAST searches against UNIVAC database, and then it trims the regions where it finds the significant matches. We'll talk about BLAST in some later lectures. Adapters, they are relatively short fragments than vectors, so they are common in NGS data though. So they are relatively short fragments, as we have said, and we can use a software called as BLAST short. So we can use this method, which is actually adopted by a software called as crumbs. It goes with exact matching algorithm called as cut adapt. What about the quality? So it is advisable to remove low quality regions. However, sometimes it may be informative. So sometimes people, they still want to keep them, but sometimes there are some thresholds. Mostly what we have seen is that the softwares like aligners, they are good if the sequencing quality is between 20 and 40. Low quality bases may be trimmed by trimmers. There are two approaches, PRINSIC or crumbs, so we can use these programs. Sometimes we don't have quality scores available, so at that time we might look into where we have large number of N's, and that can give us an idea that sequencer was not decisive what nucleotides are there, so it placed over there. So maybe we can see these N's and we can simply trim them. Duplicates are the are where we have those reads which are read twice, so they can be coming upon, they depend upon the depth of the sequencing and the type of the libraries which we construct and the sequencing technologies we are using. So that can be the factors which may give rise to these duplicates. They also arise due to PCR errors, and they are common in 454 systems. And when we are doing those mate pair sequencing in Illumina, we also see them. Sometimes we can have a double signal, so these kind of duplicates are called as optical duplicates, so they are also common in Illumina. So these signals which generate those sequences, they are read twice, so that can be recognized as presence of close to one another. So if we are seeing the similar reads close to one another, we suspect there is some optical duplicate. What duplicates might do? They calculate skewed allele frequencies in SNP calls. So we will see later on in SNP identifications. So since we are comparing the frequency of changed alleles and we are comparing them with the reference, so they might skew that pattern. They can also report the false expression profiles. So we are calculating the total messenger RNA produced by some gene. So if we have duplicates, obviously we are doubling those numbers, so it's not good. And sometimes in a sequence assembly software, they can give a wrong impression to the assembler that that assembly is correct. So they are kind of also interfering with the assemblies. Duplicate filtering might have though some issues. What happens if we have the true duplicates? What happens if we have the diploids? Since we know there are two sets of chromosomes in diploid individuals, so identifying a true duplicate from a wrong duplicate might be a cumbersome task then. Low complexity regions are where sequencers are indecisive, so these regions can have some issues. So large number of n does not mean anything, so that can create issues for the assemblers. There is a software called as DUST under NCBI, and within the BLAST, so that can mask those low complexity regions. So whenever you are running the BLAST under NCBI, it gives you the option where you can mask those low complexity regions. So they will not then will be used in the alignments. There is another software, crumbs, that also filter these low complexity regions. What about contaminants? So contaminants can be due to sample preparations. What to do with them? What kind of contaminants are there? For example, mitochondrial and chloroplast genomes in the genomic samples. There can be ribosomal RNA in transcriptomics. So whenever we are doing this RNA-seq technique, since ribosomal RNA is produced a lot in the cell as compared to the other RNA, so obviously if it is still there, it will take large number of sequencing resources, and that sequencing is just a wastage. So since we are only interested in messenger RNA, so obviously ribosomal RNA might contaminate those messenger RNA studies. There may be some pathogens in the infected samples. So for example, you are getting the expression profile of an individual, but we are getting the RNAs coming from some microorganisms, some bacteria which is sitting there, or some viruses. So contaminants can be removed by careful and skillful extractions. Obviously extraction protocols are where you can work a lot, and you can find them in the sequences by BLAST searches. And obviously crumbs, there is a software that can filter these contaminants. So what so far we have seen is that data quality is an important issue to deal with in NGS data analysis. Vectors, adapters, low complexity, quality, contaminants and duplicates, all of them, they pose specific challenges to the NGS data analysis.